Hello dear students, today we are going to learn about the Wilson's disease. Wilson disease is a metabolic disorder. What is it? It is a metabolic disorder. Metabolic disorder in which there is excess amount of copper is accumulated in our body. And due to which there is toxicity of copper leading to various symptoms. Also, Wilson disease is a hereditary disorder and it has a autosomal recessive inheritance. So both parents should be carriers. Incidence of a Wilson disease is 1 in 30,000 live births worldwide. And commonly affected age group 3 years to 50 years. Now little bit about the history of Wilson disease. In 1912, Dr. Wilson described this disease as a lenticular degeneration with liver cirrhosis and also Dr. Wilson noticed that this disease is a hereditary disorder. In 1902, KF ring was already described. In 1902, KF ring was already described, and in 1956, D-penicillamine was used as a chelation therapy in Wilson disease, and in 1961, role of zinc was identified. Role of zinc in the treatment of Wilson disease was identified. Late in 1993, first time genetic study shown the responsible mutations in the gene. In 1993, genetic studies identified the genes responsible for manifestation of Wilson disease. Now we will see copper metabolism. Usually 2 to 5 milligram per day of copper is required for our body. When copper along with food enters into the duodenum and early part of jejunum, from where copper is absorbed into the circulation. So absorption site is duodenum and early part of the jejunum. In this absorbed copper then transported to the liver, where this copper is incorporated into the ceruloplasmin, and this combination copper with ceruloplasmin is transported to other organs for its further utilization. Excess of copper which is absorbed is resecreted into the intestine. Copper present in the diet, almost 20% of it is absorbed from this duodenum and jejunum, and rest 80% is excreted through the feces. And this excess copper which was resecreted into the duodenum, this also is excreted into the feces. Usually there is very negligible quantity of copper which is excreted through the urine. or negligible amount of copper is excreted through the urine. Now we will see how this copper gets incorporated into the ceruloplasmin. So for that we have taken a single hepatocyte. Along hepatocyte there is a sinusoids on both sides, and on one side there is biliary canaliculi. This is the nucleus and these are the Golgi apparatus. Copper absorbed through the gut into the circulation enters the hepatocyte through a special transporters called as copper transporter 1 or CTR1. So copper is transferred from circulation into the hepatocyte through this copper transporter 1. Now this copper is transferred to the Golgi apparatus with the help of a special transporters called as ATP7B. These are the special transporters required for the transfer of copper from cytosol to the Golgi apparatus. So thus copper ions are transferred into the Golgi apparatus. In Golgi apparatus, apoceruloplasmin is already present. Now this copper ions binds with apoceruloplasmin to form ceruloplasmin. Apoceruloplasmin is a precursor enzyme, and this ceruloplasmin, or also called as holoceruloplasmin, it is a complete enzyme. So for the maturation of enzyme, copper ions are required, and this ceruloplasmin or holoceruloplasmin then secreted into the circulation, from where it is transferred to other body parts for the utilization or its further function. Now the copper which was in excess in the Golgi apparatus, again it can be excreted into the bile with the help of same ATP7B transporters. So the excess copper is again excreted through these vesicles into the biliary canaliculi, and from here it is excreted into the intestine and then into the feces. In Wilson disease, there is mutation in a gene present on chromosome number 13. Chromosome number 13 on its long arm from Q14 to Q21. And the gene present on this locus is required for the production of ATP7B. When there is mutation in this gene, ATP7B protein cannot be produced. When ATP7B is absent, then there are two factors which are affected. One, the copper cannot be taken into the Golgi apparatus to incorporate it into the apoceruloplasmin to form ceruloplasmin. This is number one. And number two, the ATP7B transporter is also required for the excretion of copper into the biliary canaliculi. So when this gene is mutated and ATP7B is absent, then copper is not transferred to the ceruloplasmin or cannot be excreted into the bile. So lot of copper is accumulated into the cytoplasm. As we know, copper is a highly reactive and it produces oxidative stress and injury to the cells. To avoid that, there are some protein which get binds with the copper and prevent those injuries. So those proteins are ATOX 1 proteins. But in Wilson's, there are too much copper is accumulated into the cytoplasm. Those ATOX 1 proteins are exhausted and lot of free copper ions are scattered throughout the cell, and this free copper ion causes hepatocyte damage. Further, those free coppers after hepatocyte injury can be transferred into the circulation and reaches to other organs like brain, eyes, kidneys, and etc., and which leads in the manifestation of Wilson disease.